Hello friends, I am Dr. Amit Dingwal, Associate Professor at SVKM's Institute of Pharmacy, Dhule. In this video tutorial, we will talk about general steps involved in AI-driven drug discovery. So these are the minimum required Python libraries or tools required to conduct AI-driven drug discovery through machine learning or deep learning. These are the main software used in a typical Python machine learning pipeline, which consists of almost any combination of the following tools. NumPy is a Python library for matrix and vector manipulation. Matrix and vector, scalar, tuple, tensor, list, they are the ways to represent input along with input features to train machine learning. Pandas are for time series. They are used to make data into data frame. For example, taking your data from Excel sheet into your Jupyter or Colab notebook. The 2D plotting libraries, Seaborn or Matplotlib, they are the library or the options to visualize data into various formats. Matplotlib and Seaborn. Scikit-learn is a world famous source for many machine learning algorithms and utilities. You have Excel sheet having X and Y data. X means input, Y means output. So directly from Excel sheet, data will be taken into Jupyter notebook through Panda data frame structure, and directly you can run your machine learning model directly on those data. So for running simpler machine learning model on your data, you need scikit-learn machine learning library. On the other hand, if you are going for analyzing huge amount of data, then you have to go for deep learning type of AI system. So for deep learning, you will go for Keras, because deep learning model cannot be run on scikit-learn. So first of all, in any type of machine learning or deep learning model training, any type of machine learning on this earth, it may be drug discovery, merchandise classification, e-commerce recommendation, YouTube recommendation, and all type of recommendation studies involving deep learning, you have to collect data and you have to bring data on a common normal platform. It is called to normalize the data. So whatever is the tool or purpose, whenever you handle a database, first of all you need to normalize the data or adjust values that are calculated on different scale to a common scale. For example, one data set may score numerically and another by a percentage. To compare the data, the values must be normalized to a common scale, either numerical or percentage. Second is to extract or to remove identifiable or measurable features or properties or descriptors from those data, from the input data. That is, a descriptor should reveal the inherent physical and chemical properties of the corresponding molecule in case of drug discovery, so that this statistical model can learn and generalize the common characteristics among the molecule that lead to the interaction between compounds and targets. After the machine learning models are constructed using the descriptors of known ligand receptor pair, interaction predictions are shaped for the unidentified ligand receptor couples. Here ligand means drug-like molecule, receptor means protein, which are responsible for causing diseases. So by providing query descriptors as input to the model. There are numerous types of descriptor both for small molecule compounds and target proteins receptor. Each have strength and weaknesses in terms of the power of representation of molecular property. So already we have discussed hundreds of thousands of descriptors can be there for each molecule, but we have to choose only those signature descriptors which are making those particular molecule effective for a particular receptor. So some most common major steps involved in AI driven or AI guided or AI mediated or AI empowered drug discovery. They are: decide which disease you are going to take into consideration. Decide the disorder. for which you want to search a drug molecule through AI operation. Decide if you are going for repurposing, that is to explore existing drug molecule for entirely new use, that is indication, that is off-label use, that is the use which is not mentioned in Orange Book, which is a record maintained by US FDA for the commercially available, that is FDA approved drugs. Or you want to go for second approach, that is de novo drug designing, that is to generate new molecule using generative type of neural network like GANs and others. Repurposing, also known as repositioning, involve exploring a known chemical structure for a new indication through some empirical approaches, logics, through machine learning models, because there is no logic which can be compared with machine learning logics. Here a known chemical structure means a drug molecule which is commercially and scientifically hit. A drug molecule which was okay during and even after clinical trial until it has been recalled from market owing to some side effect or adverse effect which might be overlooked during approval process or during clinical trial. A drug not hit the market owing to other reasons. A drug candidate under investigation for other indication. A drug candidate which could not see prescription owing to failure in any of the stages of drug discovery marathon for a particular disease treatment. Then choose the database which you are going to use to train, validate, and ultimately deploy your machine learning models for either de novo drug design or for finding a molecule from existing databases for a new indication, that is repurposing or repositioning. Some of the mostly used databases are Campbell, DrugBank, Zinc15, PubChem, etc. It is said that around 72 million molecules are there in general to explore from. Drug discovery usually revolves around small molecules. Exceptions are protein drugs like biologicals, insulins and other hormones, monoclonal antibodies, etc. 72 million is just the size of a specific database. The actual number of small molecules is estimated to be between 10 to the power 60 to 10 to the power 200. Synthesizing and analyzing a single molecule in the lab may cost dollars and months, if not years. And see the number of total molecules. A small molecule means molecule that can easily get through a cell membrane, that is anything smaller than a nucleic acid. Convert all chemical structures into machine readable format through various molecular representation technique. Already we have seen in earlier videos. Advanced sort of molecular representation tools and types are there which not only learn about chemical structures, bonds, atoms, but also about entire chemical system, that is its neighborhood, vicinity area, which is required for a drug to be fit for diabetes treatment, for example. And this job is done very nicely by RDKit, a cheminformatic library, in a Python friendly manner. Remove all unwanted data like outliers, duplicates, invalids, etc. In entire journey at various steps, you have to go for law of elimination, that is to remove unwanted structures, substructures using Tanimoto index and other similar parameter. This causes removal of panes. Extract feature from datasets using open source chemical software like RDKit. Choose the machine learning model for simple classification, regression or clustering sort of tasks, or all as ensemble group of more than one algorithm for same input for same problem, to better understand the prediction of machine learning model before jumping to conclusion while handling hundreds of thousands of drug molecules from database mentioned in previous slide. Apply a machine learning model depending on your workflow pipeline. Say for example, recurrent neural network to a large number of chemical structures represented in the smile string to obtain generative model which learn the probability distribution of character in a smile string. Train, that is teach, that is to supervise machine learning task, that is make your chosen machine learning model to understand what is required for a molecule to be an anti-diabetic molecule, for example. Training starts by teaching model through mathematical expression, statistical tool about most important features or identity function which makes an antidiabetic molecule an antidiabetic molecule, and separate from those molecules which are not and which can never be antidiabetic molecule because of their chemical structure or rules like Lipinski's docking, binding data and others. Depending on type of computation tool like processes, GPUs, type of machine learning model, type and pattern of data, training a machine learning model may take days, months also. For small data sets, small means less than 1000 molecule. It is possible to train models within a few minutes on a standard laptop with CPUs only. However, for larger data sets, GPU is required for significantly faster training. While training the model, it is important to use hundreds of drug molecules or drug-like molecule from various sources to teach model about variety of positive and negative compounds. More the variety of molecule, less the chance for model to be called underfit or overfit. Model should be exposed to lot many type of chemical structures during training to make it a successful generalized machine learning model for searching anti-diabetic molecule post its testing. Successful training of the model is followed by validation and testing of the model before deploying the model for predicting the output of for a problem using unknown data but for same problem. Here problem means to identify a drug molecule from database or to make a new drug molecule through generative type of model, that is de novo drug design for its use in diabetes treatment. Lot many formulae like error, cost function, sigmoid activation function, ReLU activation function, sort of things, scoring function, binding score are involved to fine tune the model to minimize the error while getting the machine prediction value which should be close to the actual output. It is called cost function. That is what is the cost function? The difference between the machine given result and the actual, that is the ground truth. So the purpose of machine learning model training and tuning and testing is to minimize the cost value. Your machine given prediction or output should be as close to the actual value as possible. Having trained the model, now having trained the model, it is tested using those molecules which have not been used during training. After training, model is now ready to be used for exposing it to huge data set to identify a chemical or drug molecule which may have the potential as anti-diabetic molecule. Here a perfectly trained machine learning model or deep learning neural network will choose molecule for diabetes treatment from existing database if it is repurposing, or it will make or generate from scratch, that is de novo drug design if you are going to use generative type of deep learning model, that is autoencoders of various type. A system does not know about mechanism of action etc. like humans. So here trained machine learning model will bring all possible structures without any ambiguity, without any bias which may be there if we are going through human inputs, which may or may not have very good similarity chemically with established antidiabetic molecule and may or may not have known mechanism of action. With this, we have summarized general step for developing a machine learning model. Thank you very much.